Well, if there are any questions on what we've done recently, we're going to do what we usually do, and we're going to go to screen sharing. And we're going to pick up where we left off the other day. We're going to be talking about prokaryotic cells and prokaryotes really for a few days now. So yesterday we talked mostly about the genetic material, that single circular chromosome that we find. And today we're going to move to some of the other parts of the cell. And keep in mind what we said back in Biology 105, when we think of cells, really the best way to think of them is a description that I read once. They're organic factories that make proteins. And then they use these proteins to carry out various activities. So that's one of the reasons why we focus so much on the DNA. That's the instructions to make the proteins. And that's the on-off switches that tell the cell when to make the proteins or when not to make the proteins, I guess, depending on your point of view. And so we talked about that single twisted up circular chromosome in the nucleoid region. And really there's one organelle that we find in the cytoplasm of all prokaryotic cells. There are a few others that if you look at various pictures, you may see diagrams of, but they're not really found in all prokaryotic cells. And the one organelle that's found in every prokaryotic cell, again, think about this idea that cells are organic factories that make proteins. Well, to make proteins, you need ribosomes. So let's look at the difference between how a prokaryotic cell makes proteins and how a eukaryotic cell makes proteins. So remember, in protein synthesis, there are two steps. There's transcription. And in transcription, we take the DNA and we use it to make RNA. And then there's translation. And in translation, we take messenger RNA and we use that to make protein. Remember, there are lots of types of RNA that we can make in transcription. We can make tRNA, we can make rRNA, make all sorts of RNA, but only mRNA gets translated into proteins. Well, on the right in our diagram here is a eukaryotic cell. And if we look at this eukaryotic cell, this dark purple oval with the little holes every once in a while represents our nuclear envelope, or if you prefer, nuclear membrane. So if we think about this nuclear membrane, notice that transcription, the process where we make messenger RNA, happens inside the nucleus. But the ribosomes, these little pinkish blobs down here towards the bottom, the ribosomes are out in the cytoplasm. And the ribosomes are too large to fit through those little holes, those nuclear pores. So the ribosomes are out in the cytoplasm. So a eukaryotic cell does transcription inside the nucleus. Then it transports the messenger RNA out into the cytoplasm to a ribosome. And out in the cytoplasm, it does translation. So that nuclear envelope is crucial in a eukaryotic cell. It separates transcription from translation. And remember, we talked about the idea that in eukaryotic cells, most of the messenger RNA that gets made actually never gets used to make a protein. 20% or less typically is used to make a protein. On the other hand, in a prokaryotic cell, almost 100% of the messenger RNA that is produced is used to make protein. Why the difference? Well, let's look on the left at our prokaryotic cell. So this kind of pink peachy oval on the outside is our plasma membrane. Now remember, we've got a cell wall outside that, and we might have a capsid outside that. We could have flagella, we could have pili sticking through that capsid. But the key thing is, if we look at this squiggly purple line in here, this is our single circular chromosome. Notice there's nothing that separates the chromosome from these little pink blobs, the ribosomes. So as soon as the messenger RNA gets made, and it's kind of a bluish line here, as soon as the messenger RNA gets made and it starts to kind of peel off of the DNA, ribosomes attach, and the ribosomes start making proteins. So here in a eukaryotic cell, we make messenger RNA, we might or might not use it to make proteins. In a prokaryotic cell, if we make messenger RNA, we are going to use it to make proteins, because as soon as we make it, a ribosome attaches. Well, remember that ribosomes are made up of two parts. They're made up of a large subunit and a small subunit. And in prokaryotic cells, the large subunit is 50S. S is Svedberg units, which is a measure of their size. And the small subunit is 30S, 30 Svedberg units. Put them together and the whole ribosome is 70S. Notice you don't add up 50 plus 30. It might seem like you would, but this really refers to when you put these particles into a centrifuge, what speed they separate out at. So this is not really an additive unit. If we look at the ribosomes, keep in mind ribosomes are made of two types of molecules. They're made of rRNA, and R, of course, is ribosomal RNA, and they're made of proteins. And in a typical ribosome, about 65% of the weight would be ribosomal RNA, and about 35% of the weight would be protein. And notice there are several different sizes of rRNA. There are several different sizes of protein. And we'll come back to that a little bit later when we talk about some other prokaryotic cells. So remember, what the ribosomes do is they read the messenger RNA, and then transfer RNA adds amino acids, which is spelled wrong. Where does it get those amino acids? Well, it collects them from the cytoplasm. When you think of the cytoplasm, think of a warehouse. It's where our cell is going to store reactants for all the reactions, including protein synthesis. And remember, we have that complementary base pairing between the codon on the messenger RNA and the anticodon on the transfer RNA. And that ensures that we get our amino acids in the correct order. So if we look at our original cell, all these little pinkish blobs are our ribosomes. This kind of orange or peach color in the background is the cytoplasm. And so notice if we think about the cytoplasm, we mentioned it a little bit the other day. It's kind of a semi-liquid, semi-solid, kind of a milkshake type of consistency. And if you look at the cytoplasm, the most common molecule in there is water. But there are lots of inorganic ions dissolved in the water. Things like sodium ions, potassium ions, calcium ions, chloride ions. What are all these different ions? Well, some of them are coenzymes that work with the enzymes. Some of them play a role in maintaining what's called the osmotic pressure of the cell, which is just the more technical way of saying they stop the cell from dehydrating. And then the ribosome and the chromosome are so big, they're not really dissolved in the cytoplasm, they're more suspended. So if you're thinking of this like a milkshake, this is like one of those blizzards or something where you've got your little M&Ms or something floating in the milkshake material. Okay, the next thing we want to think about is this little peach colored line, the plasma membrane, sometimes also called the cell membrane. And this surrounds the cytoplasm and all the organelles that are in the cytoplasm. And the plasma membrane is crucial. If the plasma membrane is punctured, cytoplasm and organelles start leaking out and the cell dies. So remember, when we think about our plasma membrane is what's sometimes called a phospholipid bilayer. And remember that when we look at phospholipids, we see this kind of golden part, which is the polar head area. And then we see these two black lines, which are the nonpolar tails. And remember that we made our phospholipids, was synthesized in the cell by taking a molecule of glycerol, which has 3 carbons, combining 2 of those carbons with fatty acids. So what's left of the fatty acids is these long nonpolar tails. And then the 3rd carbon in glycerol, so 2 of them are combined with fatty acids. 1 of them is combined with a phosphate group. There's usually also an amine group up here, but not always. So if we look at this plasma membrane, this would be taking a slice through it, kind of like slicing through, if you would imagine that our cell is a beach ball. Outside, we have the outer environment, and bacteria cells live in environments that are mostly fluid, so water. So notice that the polar head of one layer is facing outward against the water of the environment. On the other side, the polar heads are facing inward against the cytoplasm. Well, remember, we just said a minute ago, the cytoplasm is mostly water. Now, one of our rules in chemistry is polar things mix with polar things, nonpolar things mix with nonpolar things, but usually polar and nonpolar things don't mix very well. So the polar heads are out against the water of the environment, and they're also on the very inner layer against the water of the cytoplasm. But most of the thickness of the membrane is the nonpolar tails, which are hydrophobic, meaning they don't mix with water. So the plasma membrane basically keeps stuff in or keeps stuff out. Water and the things that are dissolved in water can't pass through the membrane. So things that start out outside, stay outside. Things that begin on the inside, stay inside. Now remember, the other type of molecules that we see a lot of in our plasma membrane are proteins. And whereas the phospholipids mostly keep stuff in or out, depending on where it started, the proteins are what move things from the outside of the membrane to the inside. So they move things into the cytoplasm, or they can move things out of the cytoplasm. About the only things that can get across the membrane without being transported by proteins are things that are nonpolar, because most of the thickness of the membrane is nonpolar. So there's at least 2 very important nonpolar molecules that all living things have to either get into the cell or get out. And that is oxygen is nonpolar, O2, and carbon dioxide, CO2, is also nonpolar. So these 2 materials can just diffuse across the membrane from where they're at a high concentration to where they're at a low concentration. So if we have a cell that's using up oxygen as a result of aerobic cell respiration, then oxygen will be at a low concentration inside the cytoplasm. So this means oxygen will diffuse into the cell. If we have a cell that's carrying out a lot of aerobic cell respiration, remember that one of the products of aerobic cell respiration is carbon dioxide. So carbon dioxide will diffuse out of the cell. A few other materials like lipids can diffuse in and out of plasma membranes, but that's mostly important in multicellular organisms like animals. There aren't too many lipids floating around in the environment of bacteria cells. So the phospholipids are primarily to keep stuff in or keep stuff out. The proteins are primarily to move things across the membrane. But proteins also serve another important function when we talk about bacteria that cause diseases and how our immune system fights bacteria that cause diseases. If we look here, we can see the outside of the cell is again up. We can see that some of the proteins, these big purple blobs, stick up outside the cell. These proteins are molecules that your immune system can recognize. So when you get a bacterial disease, if your immune system fights off that disease, in the process of fighting it off, your immune system learns to recognize these proteins. And then the next time you're exposed to that disease, you don't get it. Why? Because your immune system responds so quickly, so strongly, and it's such a long-lasting response. You know, if you ever had, an illness like the mumps when you were a small child, after you get the mumps once, if your immune system is strong enough, you usually don't get it again. Now, if your immune system is weak, you might not remember how to recognize the proteins, so you might get it again. So these types of proteins have been in the news a lot lately, not with bacteria, but with viruses. You've all seen those pictures of the virus that causes COVID-19. Guess what? Those big spikes that you see on the outside of that virus, those are proteins. And these proteins are important in a bacteria or a virus, not because it's what our immune system recognizes. That's important to us. But for bacteria and viruses, these proteins are important because those proteins allow a bacteria or virus to attach to one of your cells. So when they talk about mutations to the COVID virus, like they've been talking about recently, mutations are always changes in the genetic material. But what scientists are worried about is that those changes in the genetic material are going to change the shape of that big spike protein, because that big spike protein is how this COVID virus enters your cells. It's also how your immune system recognizes the virus. And it's also the basis for the vaccines that are being given to people. So if there's a mutation, if there's a change in the genetic material, and it changes the amino acids in that protein, that might mean that your immune system wouldn't be able to recognize the spike protein anymore. So it might mean even though you've already had COVID-19, you could get it again. Or it might mean that you've had the vaccination and the vaccination doesn't really help you. So we'll talk more about that when we talk about viruses. But keep in mind that these proteins have 2 points of significance. They're important for bacteria that attack cells in other organisms because they're the point of attachment, but they're also important for the immune system. This is what the immune system recognizes to fight that off. So we want to keep that in mind. So we've talked about ribosomes, the cytoplasm, the plasma membrane, the chromosome. And really, if we look at the other parts of bacteria cells, there's one other main part that is found in all bacteria cells. And this is the part that on this model is shown in the black. And this black part is the cell wall. Now, notice there's a thin brown line inside this. This is the plasma membrane. And notice that this plasma membrane has an indentation in it. This is called an invagination. And an invagination is where something bends inward. And this invagination is called a mesosome. Now, remember when we talked about aerobic cell respiration and photosynthesis last semester? One of the things we talked about was a process called chemiosmosis. And remember, in chemiosmosis, the cell transported hydrogen ions across a membrane. And then when the hydrogen ions moved back where they started, the cell was able to use that movement to produce ATP. In a lot of bacteria cells, chemiosmosis happens in these mesosomes. So if they carry out aerobic cell respiration, or if they carry out photosynthesis, there are some bacteria that are photosynthetic. This is where that occurs in these mesosomes. All bacteria cells don't have it, and they don't have to have a mesosome to carry out aerobic cell respiration, but it definitely helps. Now, when we look at the cell wall, this black line around here, keep in mind that the cell wall, excuse me, is continued to be considered to be non-living material. The plasma membrane is living and everything inside it, the cytoplasm. But keep in mind, the plasma membrane and everything inside it is only living if you consider it as a whole. Ribosomes aren't alive, chromosomes aren't alive, the cytoplasm isn't alive. All of these combined inside a plasma membrane are alive. The cell wall isn't considered to be alive because if you're very careful, you can pull the cell wall away from the plasma membrane. And if you don't damage the plasma membrane and you have the cell in the right type of osmotic conditions, if the concentrations of different solutes are balanced correctly, the cell can live. So why is the cell wall there? Well, keep in mind, since the cell wall is non-living, the cell wall is actually made in the cytoplasm. And then it's transported out through the plasma membrane. Now in bacteria, the cell wall is made mostly of some material called peptidoglycan. So if you think about the name peptidoglycan, remember peptides or polypeptides was another name for a chain of amino acids. So peptido refers to chains of amino acids. Glycan comes from the same root as glycolysis or glycogen, and glyco refers to carbohydrates. So the cell wall in most bacteria is made of peptidoglycan, long polysaccharides, and the long polysaccharides are connected and held to each other by short chains of amino acids, peptido. So that's where the name comes from. Now we want to keep peptidoglycans in mind because later we're going to look at plant cells, which also have a cell wall. And we're going to look at fungus cells, which also have a cell wall. And we're going to look at archaea cells, which also have a cell wall. None of those cell walls are made of peptidoglycan. So chemically, if I gave you a chunk of cell wall and you didn't know what type of cell it came from, we could do a chemical analysis and we could figure out what type of cell it came from, depending on what compounds were in it. Now, not all bacteria, but a lot of bacteria have this kind of pink layer outside the cell wall. And this is something called a capsule or a slime capsule. And it's made of something called a mucopolysaccharide. So think of mucus, and polysaccharide, again, is a big carbohydrate. And really, if we look at the cell wall and the capsule, which is sometimes called a slime capsule, they have very similar functions. They both protect the cell. Like the cell wall, the slime capsule itself is not considered to be alive. So if we think about the slime capsule and the cell wall, why do you have all this non-living stuff outside the plasma membrane? For protection. The cell wall and even the capsule are thicker and they're stiffer than the plasma membrane is. The plasma membrane is lipids, so it's flexible. Think of a drop of oil floating in a frying pan. The cell wall and the the slime capsule have a fairly distinctive shape. So one of the things they do is they give a bacteria cell its shape. The other thing they do is they protect the bacteria cell. some antibiotics that we apply can't get through the capsule and the cell wall. And most antibiotics have to get into the cytoplasm to have an impact on the bacteria cell. Not only do they protect the bacteria cell from antibiotics, but they prevent the bacteria cell from absorbing so much water that it swells up and bursts like a balloon that you put too much air into. Remember, we looked at red blood cells going through plasmolysis, where they absorb so much water, they just popped. That doesn't happen to bacteria cells because the cell wall is so thick and stiff and strong. When the cytoplasm starts to absorb more water, eventually the cytoplasm is pressing out against the inner edge of the cell wall, and the cell wall doesn't have any give to it. So the cytoplasm can't absorb any more water at that point. And so it stops the cell from absorbing too much water. It protects the cell from antibiotics. It protects the cell from viruses. There are viruses that infect bacteria. Some of them can't get through the slime layer or the cell wall. And it also protects the cell from friction. Some bacteria cells can move. And just like in a moving car, you have friction. That's why your tires eventually wear down and you have to replace them. Cell wall and the capsule continually have to be built back up because in bacteria that move, they're constantly wearing down. So the capsule is something that is outside the cell wall but has similar functions. Now, I said that the cell wall helps to give the bacteria cell its shape. And when we look at bacteria cells, there are three basic shapes. Some people say that there's four. Two of the shapes everybody agrees on. One shape sometimes gets subdivided. So there are cells that are called coccus cells. Coccus cells are spherical. Now, keep in mind, these cells might look round, but really they're spherical because although they are very small, they're three-dimensional. So we don't really just want to say they're circular. And there are some coccus bacteria that cause illnesses that you've heard of. You might not realize they're coccus bacteria, but you've probably heard of something called strep throat. One of the causes of strep throat is a bacteria called Streptococcus. So the shape of the cell is in the name. There's another coccus-shaped bacteria called Staphylococcus. So you might have heard of staph infections, or you might have heard of MRSA. The S in MRSA is Staphylococcus. There are some cells that are kind of rectangular, but they usually have rounded ends. So, you know, they're kind of shaped more like a hot dog than a rectangular box. These are called bacillus-shaped cells. So one bacteria that you've probably all heard of is something called E. coli, Escherichia coli. E. coli is a bacillus-shaped bacteria. Although E. coli is a really short bacillus, it's maybe about, I don't know, a third the length of one of these green bacillus cells. In fact, it's so short that it's almost a coccus because, you know, since the ends are rounded, if you get too short of a bacillus, it looks more like a coccus-shaped cell. The other basic shape, and for this, you have to keep in mind that this really is three-dimensional. If you look at some of these purple squiggles here, you can see that they twist back and forth. You might have seen some pasta noodles that show this kind of twisting pattern. This basic shape is called a spirillum shape. Now, whereas coccus bacteria are all basically round, bacillus bacteria are all vaguely rectangular, spirillum bacteria have a little more variety. Some of them have a couple of folds, a couple of twists, depending on how you want to look at it. So they look kind of like the letter W or M. But some of them just have one twist. So they look more like a comma or the letter C. And some of them have 5 or 6 twists, so they look like a squiggly line. If you've had a math class, something like maybe a sine wave or a cosine wave. Some people, if we have a really short spirillum shape so that there's only one twist, so that it looks like a C or it looks like a comma, they call that a vibrio shape. We're not going to worry about whether there's really 3 shapes or 4 shapes. We'll let the microbiologists sort that out. But keep in mind, if you're reading something about bacteria, yeah, you might see one reading that says there are 3 shapes, and you might see something else that talks about 4. So we're good either way. If we look on the outside of our cell, let's go back here for a minute, we sometimes have some structures that stick out. We have some things called pili. If we just have one of them, it's a pilus. US is singular, I is plural. Keep in mind that's because these are derived from Latin words. A lot of science terms are. And in Latin, when you have a word that ends in US, you make the plural by dropping the US and adding I. Pili are what bacteria cells use to attach to other stuff. And really, they can attach to 3 types of things with these pili. They can attach to a host cell. So bacteria that cause some infections in the human body use their pili to attach to your cells. They can also attach to something called the substrate. The substrate is just the surface that they live on. So some of the bacteria that cause cavities, for instance, attach to the enamel on your teeth. Now, the enamel is not really living cells, it's produced by cells, but the enamel itself is not alive. So those bacteria can use their pili to attach to this substrate, to this enamel. And some bacteria use their pili to attach to other bacteria. That's how they reproduce. Well, no, I'm sorry, I really stated that incorrect. I want to correct it. That's how they exchange DNA. But technically, bacteria don't reproduce with each other. So some microbiologists give pili different names, depending on whether they use it to attach to the host cell that they're infecting, or another bacteria cell to exchange DNA, or to a substrate. We're just going to call them pili. If you take microbiology, you'll get to learn some of the distinctions between the different types of pili. Some bacteria have what's called a flagellum, or some have more than one. This is another word that has an irregular plural. If there's one, it's a flagellum. If there's 2 or 3 or 23, it's flagella. It ends in A instead of UM. And the flagellum in bacteria cells works like a propeller on a boat motor or on an old airplane that isn't a jet. It basically spins in circles. And if we look at a flagellum, we'll talk about flagella a little bit more when we talk about eukaryotic cells, because there are eukaryotic cells that have flagella. Every sperm cell has a flagellum. But the flagellum in eukaryotic cells is made of different chemicals, and the chemicals are organized differently. And when I say chemicals, keep in mind we mean proteins. So in prokaryotic cells, flagellum are made of a protein called flagellin that ends in an ion. In eukaryotic cells, flagella are made of something called tubulin, the same thing that's found in the microtubules that you can see during mitosis and meiosis, and the same thing that's found in the cytoskeleton of cells. So notice that a lot of cells don't have pili, a lot of cells don't have flagella, a lot of cells don't have a capsule. But if we talk about the chromosome, if we talk about the ribosomes, the cytoplasm, the plasma membrane, the cell wall, that's something that's found in almost all bacteria. Keep in mind, it's hard for us to say all of anything when we're talking about nature. There are a few weird bacteria that don't have cell walls, for instance. There's not anything that doesn't have a plasma membrane that I'm aware of, but there are some bacteria cells that have two membranes. They have an inner membrane and an outer membrane. There are some bacteria that have two different cell walls. So there is a lot of variation. There's so much variation, in fact, when we look at bacteria, they show more variation in terms of their cell structure or their metabolism. Just within bacteria, there's more variation than there is in all the eukaryotic organisms combined, plants, animals, fungi, the things that are none of the above. So there are a lot of different bacteria. So we want to keep that in mind. Now, when we look at bacteria, keep in mind that the oldest fossils that have ever been discovered are bacteria. And we believe that the Earth is about 4.6 billion years old, as best we can tell. And we have evidence of bacteria going back 3.5 billion years. And there's some indirect evidence of bacteria. We don't have the cells, but we have something that the cells made going back around 4.2 billion years ago. So not very long after the Earth formed. If we look at the classification of prokaryotic organisms, everything that had a prokaryotic cell was originally classified together in one group. The group had different names at different times. But in the late 1970s, a professor at the University of Illinois, who was studying bacteria that today we classify in archaea, started noticing that the archaea had several differences from the organisms that are in the domain bacteria now. And he was actually the one who proposed the system of domains. And he said there are three main groups. There are eukaryotic organisms, and there are bacteria, which have prokaryotic cells, and there's archaea, which also have prokaryotic cells. So originally, everything in the domain bacteria and the domain archaea was classified together until about 40 years ago. And at that point, it was recognized that archaea were so different, they shouldn't be grouped with regular bacteria. And in fact, today, the evidence suggests that archaea, even though they have prokaryotic cells, are more closely related to eukaryotes than they are to bacteria. So we'll see what we mean by that later. Now, when we think of bacteria, there's not very much to look at. So how do we divide bacteria into categories? Well, it's usually done through two different ways. We divide them into categories according to how they react when we put a stain on them and some metabolic tests that we can do. So one of the main tests that we use to divide bacteria up into groups is we put a stain on them, then we put on a chemical that removes the stain, then we put on another chemical. Some of the bacteria hold on to the stain, and they're bacteria that we call gram-positive. Some of the bacteria lose the stain and pick up the second stain. They're called gram-negative. Now, I'm not sure biologists realized this when they first started studying bacteria, and they put gram-positive bacteria and gram-negative bacteria into different categories. But the very outer part of the cell is that cell wall and maybe the capsule. And whether bacteria are gram-positive or gram-negative, that really reflects the structure of their cell wall. And when you get down to the structure of their cell wall, guess what? The cell wall is built by proteins. It's built by enzymes. So this means that when we look at the cell wall is kind of an indirect way of looking at the DNA. Keep in mind, when scientists first started classifying bacteria as gram-negative versus gram-positive, we couldn't do genetic sequencing, so that wasn't really an option. But as we've started to analyze the DNA of bacteria, we've discovered that the genetic similarities almost always parallel the similarities that we can see in how they respond to dyes. So let's look at another dye in a little more detail. Let's look at something called an acid-fast stain. And again, this is a stain, and the color we're going to see at the end of our stain depends on the chemistry of the cell walls of the bacteria that we're looking at. So we're going to add something called a primary stain, which in this case is something called carbofuchsin. And carbofuchsin is kind of a pinkish red color. And then we're going to add something that can dissolve carbofuchsin. It's called a decolorizer, and what we add is acid alcohol. That's why this is an acid-fast stain. And if we look at acid-fast bacteria, they keep the carbofuchsin that they've already absorbed. Bacteria that aren't acid-fast lose their carbofuchsin when you add the decolorizer. So this top line, we have acid-fast bacteria on the left, we have non-acid-fast bacteria on the right. This top line is what we see after we add the carbofuchsin. Now we add the decolorizer. Acid-fast bacteria still have their reddish pink color. Looks more red here, but in real life, it's usually a little more pinkish. Non-acid-fast bacteria lose their carbofuchsin, so they're clear. They don't have any stain anymore. Then we're going to add something called methylene blue, which is kind of a blue-purple color. Acid-fast bacteria are already stained with their carbofuchsin. They don't pick up methylene blue, so they're still reddish pink. But the non-acid-fast bacteria, there's no stain stuck to their cell walls. They pick up the methylene blue and they look blue. So when we look under a microscope, we can see some blue cells and some pinkish cells. So if we go back to our gram-positive stain and our gram-negative stain, this is similar, except our primary stain, the first stain we put on, is kind of a deep purplish color. Then we use a decolorizer, and then we add a pinkish color. So gram-negative bacteria lose their purple color when we put on the decolorizer, and then they pick up the pink. Now, the value of these, as I say, is that we can look at the cell walls, and it turns out to be a very good indication of if they're closely related genetically. We can also use something called an endospore stain. Some bacteria make endospores, and on here they're stained with kind of a green color. That's the type of stain we normally use for this, the one that shows spores the best. And if we look at endospores, endospores turn out to be very important in human biology. When environmental conditions deteriorate around a bacteria, some bacteria are killed, but bacteria that can make endospores start producing these spores. Now, endospores is kind of like going into hibernation for a bacteria cell. What they do is they build an endospore and they put their chromosome inside the endospore, and they put a ribosome or two in there, and they put some messenger RNA in there, and they put some protein and a little bit of cytoplasm. And the endospore has a much, much thicker cell wall than the regular bacteria cells do. So the endospore can tolerate heat and acids and all sorts of conditions that would kill the cell. So the spores can sit around, and then when conditions improve, the spores can start growing again because they've got a chromosome, they've got some ribosomes to build proteins. You know, they've got a few proteins, spare amino acids, some things like that. So it's kind of a starter kit for the cell to start growing again. Now, you might wonder, why is this important for bacteria? Well, for one simple reason. What bacteria are going to do is this allows them to survive conditions that would otherwise kill them. Okay, that's great for bacteria, but why do we care? Because some of the bacteria that produce endospores also cause food poisoning. And this is why if you're making food at home, if you're canning your own food or in industrial processes, this is why you have to heat food to a certain temperature for a certain length of time. You have to make sure that you treat the food so that you kill the endospores. If you don't, you might not have any bacteria growing on your food. So you would look at your food and you would think it's clean. But if you have spores in it, those spores can potentially start growing. So when you have canned goods, one of the things they tell you is, when you open canned goods, you know, before you open them, you always want to look and see if the sides are bulging. If the sides are bulging, a lot of times that means you have bacteria that have started growing inside the tin can. As they're carrying out respiration, they're producing carbon dioxide or some other gas as a waste product. And that gas is what's causing the tin can to bulge. So you want to find out about this because you don't want to eat that. There are some types of food poisoning that are relatively mild. You might feel uncomfortable, you might have diarrhea, you might throw up. But there are some types of food poisoning that are actually poisonous and will kill you. So every once in a while you hear about people who get food poisoning dying. So it can happen if you eat food that's been sitting out for a long time, but it can also happen if you eat canned food or food inside a plastic wrapper if it wasn't sterilized properly before it was put into that container. Now, keep in mind that bacteria cells never reproduce by mitosis or meiosis. Mitosis and meiosis only happen in eukaryotic cells. Prokaryotic cells reproduce mostly by a process called binary fission. We'll talk about one other type of reproduction a little bit later. And keep in mind, when we think about living things need to do two things to be successful. Ultimately, they need to reproduce, but first, they've got to survive long enough to reproduce. Well, there are some bacteria that can reproduce after 20 minutes. So when we have one bacteria cell that splits in half, that divides by binary fission, 20 minutes later, at least if they get enough nutrients, those two cells can both split and divide in half. What this means is in 48 hours, if this original cell and all the descendants survived, dividing every 20 minutes, even though bacteria are teeny tiny, that would weigh as much as the entire Earth. So obviously, most of these descendants don't survive. But keep in mind, this is a good example of something that we talked about with Darwin's ideas on evolution. Remember, one of the things that Darwin said was every type of living organism produces more offspring than can survive. So we're about out of time, so we're going to stop here for today, and we'll continue talking about bacteria and also archaea tomorrow. Are there any questions about what we've been discussing today?
No.
Keep in mind, if you don't have questions, that's fine. But if you have questions, feel free to interrupt as we're going along. I'd rather have you ask some questions than feel like you're lost as we go along. Okay, later today I will post the big set of evolution questions, and I've started giving back some of the other assignments that people ask for. So if you check later today, you should have those. If you don't see them later today, send me an email and I will send them to you then. If there's no questions, I'll let you go now. If you have some questions, we'll go ahead and see if we can answer them now.
Have a good day.
You too. I'll talk to you tomorrow.